Good evening, dear colleagues. Please write down into the chat. Can you hear me well? So we are waiting for the rest of the people and we'll start in 5 minutes past 6. So, guys, can you hear me well? Please write into the chat. Yes, please, if you have any question, just write it and just write them into the chat, and I will ask and I will answer them in the end of the session. Any questions regarding COVID-19 medicine, vaccines, diagnostics, and anything related with COVID-19, I will try to answer your questions. Good evening, Sadia. Good evening, Kate. Nice to meet you. So we are waiting for a couple of minutes for the rest of people that can join us to this session. Good evening, Akash. Good evening. I see that the audience is much more active than the Russian one when we made session last week. Hi everybody, hi. We'll start in 3 minutes. 2 minutes left. So let's start our webinar. First of all, let me introduce myself. My name is Bogdan. I'm a medicinal chemist. I have graduated from Moscow State University. My specialization is medicinal chemistry and organic chemistry. I graduated last year from a Department of Chemistry of Moscow State University. My master degree thesis was dedicated to castrate-resistant prostate cancer treatment, the development of new anti-tumor agents, especially the androgen receptor inhibitors. So I have been working in silico medicine, the Hong Kong-based company, for 3 years, and now I am leading the project dedicated to the development of new antivirals, especially for the COVID-19. We started our COVID-19 program in January, and I led the project in March. So we are developing new antivirals, new small molecules that can treat COVID-19, that can inhibit the replication of SARS-CoV-2 virus. So first of all, I want to tell you about the vaccines and small molecule medicines, that these are definitely very different areas of treatment for COVID-19. So first of all, what we can tell about vaccines and drugs. Vaccines, that's are something that can prevent the disease, not to treat already infected people, but to prevent people from being infected. So the main aim of the vaccination is the strategical. So we are focusing on the collective immune response. So as much people will be vaccinated against the COVID-19, less probability of future epidemic or pandemic will be. On the other hand, drugs, they can treat the disease for already infected people, and the main aim is tactical, so we are reducing the number of severe cases and reducing the hospitalization period. So the main aim is not to prevent the disease, but to treat the disease for already infected people. How many vaccines are being developed nowadays? Every country, every developed or developing country with a good health system, healthcare system, is developing one or two or more, much more vaccines. So these vaccines could be separated into the different categories. The main category being developed is a protein-based vaccine. So we are developing not, sorry for this kind of disruption. So main category is a protein-based category. Here we need to look into the life cycle of coronavirus, and to look at the main proteins that are connected to the virus life cycle. So here we can see the green one, spike protein, the essential protein for binding to the human lung cell, and the spike protein is very important for life cycle of virus. So without binding to the host organism cell, the viral particle cannot be included and proceed the whole virus cycle. So the protein-based vaccine is based on the protein particles. For example, for the spike protein, you can see here is orange, and then here is a green one. So immune response is based on recognition of the spike protein based vaccine. So first of all, immune system recognizes the spike protein, and then when the immune system is facing to the live organism, to the live virus, with these spike proteins it kills, because it has already developed the immune response to the spike proteins. Another very interesting category of vaccines is a nucleic acid vaccine. So it's mainly based on DNAs and RNAs, and the main idea is to develop the immune response for the proteins being produced from the translation. So you're you're giving the host cell the mRNA, so matrix RNA, and they undergo the translation and the producing of protein. Mass majority of mRNA vaccines are based to produce spike protein, we already know, and while the host cell producing this protein, the spike protein, from the spike transcript, mRNA transcript, it develops the immune response. And after the infection of the live aggressive strain of coronavirus, it's already known that, okay, that is the product of the transcript from mRNA. I know it's a spike protein, and I will kill the whole virus. And that's the main idea of mRNA vaccines. Unfortunately, there is no known effective vaccine already in the market. So we mainly know the virus vaccines. So virus are the oldest vaccines we know. So even the first vaccines against the pox, very severe disease from past centuries, it has been killed by the virus vaccines, so inactivated and weakened viruses. So we are just put the virus into a very bad environment. It going be inactivated and weakened, and these are the primary usage of all vaccines against polio viruses, pox viruses, and the vast majority of viruses we know. However, it's not very efficient, could be not very efficient for new viruses, and as you can see from this chart, viral vectors, nucleic acid and protein-based vaccines are much more popular. Even there is no precedence for a new type of vaccines. But if they will succeed, it will be the new era of the vaccines for viral vector and nucleic acid. So for viral vector vaccines, the mechanism is very similar for nucleic acid. So you are putting the genomic information into the other virus, for example, adenovirus. Adenovirus, it's not very pathogenic for human cells, but you are putting the genomic particle of the coronavirus into the adenovirus, and therefore it will replicate the particles very similar to coronavirus, proteins very similar to coronavirus, and if the immune system in the future face to the real coronavirus, it will be ready to kill because of the developed immune response. Regarding other categories, so other categories are very interesting. You know, there are some kind of speculations if the vaccine against tuberculosis, so the known BCG vaccine, can be very effective against the coronavirus. So there are some speculation that the countries with the developed program of the for preventing tuberculosis based on the BCG vaccine are less suffered from coronavirus. So other vaccines are vaccines against the tuberculosis and the poliovirus that could be effective against the coronavirus. So that was all for vaccines. What are for drugs? So the two main strategies for drugs are repurposing and the novel drugs development. So repurposing, if you're taking the known drug that's already in the market and you are trying to treat with this drug the real disease of coronavirus. So you are trying to, for example, use the anti-human immunodeficiency virus therapies against coronavirus, or you are taking the anti-hepatitis treatments against coronavirus. The main idea is to develop very rapidly known drugs with a known drug safety profile. So the main idea is to use already known safe drugs, because the most time costful step in the development of drugs is identification of safety profile. So we do not know for the novel drugs if they are safe or not. They could be effective against the viral protein, but they can be very toxic for the organism, starting from the mice to the humans. So the main idea for developing novel drugs, they could be much more effective than the repurposed ones. So we need to focus on the novel drugs if we want to make something much more effective against the novel disease. But if we need to just save people starting from the first months of the disease outbreak, we need to repurpose. So we are in cellular medicine are mainly focused on developing new drugs. But first of all, we need to finish discussing repurposing. What are the main repurposing drugs known from the clinical studies, from the clinical usage? So first of all, we need to talk about remdesivir, a very interesting drug, firstly developed against the Ebola virus, Ebola disease, Ebola fever, and it targets the viral RNA-dependent RNA polymerase. So very essential protein that move just to the virus cycle. Here you can see the viral polymerase that replicates the genomic information of the virus. So it's a very essential protein to the in the virus life cycle, and it's already approved in the USA. It's the main drug now used for the severe cases of the COVID-19. The second very interesting drug is lopinavir-ritonavir. Firstly, targeting human immunodeficiency virus, so it possibly inhibit the viral main protease of the SARS-CoV-2, and now it's involved in the phase 3 clinical trials. The third one drug is a favipiravir. It's the first drug approved in Russia, and in Russia it's called avifavir. It's not a Russia development drug, so the initial country that developed this kind of treatment is Japan, so it's a Japanese drug, and the Russian chemists developed a new synthetic route to get this compound and developed it's in like a new treatment of COVID-19, provided the clinical trials for this compound. So we will see in the future, is the drug effective or not against the COVID-19, but the clinical trials were very fast. So I believe that it's not a good idea for getting new drugs so that very fast manner. So we will see, maybe these drugs could be very effective, and it's as the remdesivir, it's also targeting RNA-dependent RNA polymerase. So a very interesting one drug is a hydroxychloroquine. So with the main defender of Donald Trump, that's making a very big PR company for this drug in his Twitter account. So this drug was initially developed against the malaria, but nowadays it's also used for as an anti-arthritis treatment. And the possible pathway that these drugs can treat the COVID-19 is the blocking of immune response, as it for arthritis that could be possibly interacts with some immune response pathways, but it's a very controversial drug and a very controversial results of clinical trials. Even the increasing of death cases after treatment with hydroxychloroquine was indicated. But you know, the World Health Organization for some time have stopped the clinical trials, and that continued once more. So I think and in the end of the year, I don't think, but I hope in the end of the year we will find the final decision if the hydroxychloroquine and its derivatives, I mean the chloroquine derivatives, are effective against COVID-19 or not. And very good results are showed by various antibodies, for example, tocilizumab or Actempra, the trade name of this antibody. It's also depends on the immune response pathways. Mainly it inhibits interleukin 6 receptor pathways, so it's also involved in the clinical trials in the phase 3. So I believe it's a very good idea to use anti-arthritis antibodies, because the main causes, causes of deaths from COVID-19, it's not a viral response, but an immune system response. And the cytokines produced in a very rapid way, and they destroys the immune system, destroys the lungs, destroys other systems of human organism. And the main idea to prevent this very aggressive immune response. So the next field I am involved is novel drugs development. So the stages of the drug development are very big and slow. In fact, it starts from hundreds of compounds, and then it goes to the preclinical stage, to the clinical trial stage, and after all stages, after more than 10 years of the development, maybe one compound will reach market and being approved, for example, by FDA, the main regulatory agency in the USA that regulates the drug market. So it's a very risky, low and slow business development of new drugs. And how can we reduce the time, the costs for example, for the first stage? So here is a drug discovery, mainly 2-3 years of the development. How can we reduce? So first of all, we would say that only very big companies can develop the drugs from the first stage. We call them big pharma companies. So first of all, there are Pfizer, for example, AstraZeneca, Novartis, Roche, Johnson & Johnson. These are very big companies that can develop the drugs from the initial stages to the market deployment. And these companies are looking for technologies that can reduce the costs, that reduce the time that we need to develop the new drugs. So generative chemistry is a very novel technology that can help us to reduce the time and reduce the costs of developing new drugs. We from Silicon Medicine are developing this very interesting and very effective technology. We are developing this technology for almost 5 years. And last year we got very interesting results. They have been published in Nature Biotechnology journal. So it was like a very loud speaking article, became one of the most popular articles from the whole history of the journal. So it became the 5th according to the Altimetric score. So what we have done, we used our generative models based on the deep learning technologies of some like artificial intelligence models. we use them to generate novel compounds targeting DDR kinase, DDR1 kinase. It's a drug target mainly focused for treatment of fibrosis. Fibrosis, you know, is a very severe disease that can be developed in the lungs or in the liver, and the main idea is to generate in a very small period of time new structures, to synthesize them and then to test them in biological studies to find out the activity against the kinase. So it took only 46 days, comparing with, for example, 2 or 3 years of the basic development of the compounds, to find new novel and very potent nanomolar compounds against this drug target. So we showed that it's very good idea to use artificial intelligence and deep learning algorithms to find new medicines, for example, treating fibrosis. And in January, we faced why not to use our deep learning algorithms, AI algorithms, to find a cure against COVID-19. So we found that there are some very interesting drug targets in the SARS-CoV-2 virus that we can use to find a treatment. So there are a lot of structural data in the protein data bank for the coronaviral proteins. So here you can see the papain-like protease, 3C-like protease, or are they called main protease. Here you can see the spike protein. Its structure is also, you can find it in the protein data bank. And we used the crystal structure for main protease. Here you can see its name 6LU7. It's available on the protein data bank, and we based our generation on this crystal structure. It took only 4 days for compound generation. We have prepared the primary publication called preprint just for 1 day, and we have started the validation process just in February. So first synthesis and then validation. Nowadays we have obtained very good and very efficient micromolar hits. I believe we will publish these compounds and these results in the next, I think, 1 or 2 months in a very good journal, maybe Nature or Science. So another attempt to generate compounds was in April and May. We have developed these compounds and reviewed using VR technologies. Here you can see the link for our website. You can see our compounds, our attempts, and how we can use VR technologies from Nano AI. So it's an American-based company. We used VR to review our compounds just in this VR set. Very interesting. I will share the link and you can see by yourself how we reviewed this compound from in silico medicine generative pipeline. Here you can see very interesting structures we have developed. So these are all potential inhibitors of the coronavirus main protease. We have published the structures in an open source, so everyone from all over the world can look at our article, look at our structures, and develop by themselves to synthesize and to validate this hypothesis of potential. We are the leaders in the generative chemistry. We have announced as most promising company developing generative chemistry pipelines by MIT. So MIT Technology Review published that in Silicon Valley, and it's not only a leader but also a pioneer in generative chemistry. So we will look at our results just for SARS-CoV-2 efforts in, I think, in 1 or 2 months. In a very good article from a high peer-reviewed journal. And here you can see the viral protease structure. Just a moment, I will show you not only this two-dimensional structure, but also, okay, so here you can see the plugin from PyMol, and we can play with the structure to show. So it is a viral protease. You can download the structure of this protease from the protein data bank. And here you can see the inhibitor of this structure published from German team. They published the results of their work in the Science, very high peer-reviewed and very authoritative journal. So the blue one is the inhibitor of the main protease of coronavirus, and it prevents of the cleavage of the viral polyprotein. So the main idea of inhibiting main protease of coronavirus is to inhibit the cleavage of viral polyprotein. So the main function of main proteins is to cleave the polyprotein after the protein synthesis into separate particles of coronavirus. So if we inhibit this viral protease with the inhibitor, we can stop the life cycle of the protein. So they have developed this and published in the Science, very, very good journal. And what we already made, very interesting idea. So we use, I will zoom it, okay. Here you can see another protein structure of viral protease. Here you can see the blue one inhibitor from PDB structure 6W63. You can download this protein structure by yourself and look and play with it. So, the main idea of generative chemistry is to use some abstraction, some very important points of inhibitor-protein interaction. Here you can see the this nitrogen atom that interacts with this white hydrogen atom. It's called hydrogen bond. So this inhibitor interacts with the protein using this nitrogen to hydrogen bond. Also, very important point is this thread butyl group that can interact with the protein as the hydrophobic group. And our generative chemistry pipeline idea is to use these very important points of hydrogen bonding and the hydrophobic interaction of this known inhibitor to generate novel compounds. So we are telling our generative pipeline, use these points to generate something novel, and I will show you what we have generated. So here you can see the novel compound. It is the different one from we have already seen. So this, the blue one. Here you can see the same bonding of this nitrogen atom to this hydrogen atom. So we use this hydrogen bond to generate novel compound. And here you can see, instead of a tetrabutyl group from the initial compound, 3 fluoromethyl. So these are fluorine atoms, and let me just use both structures in the same picture. So here you can see that these are different compounds. This is the first one, the one published before using the crystal structure, X-ray diffraction data, and here are our novel compound that was generated using our deep learning artificial intelligence algorithm. So the main idea to tell artificial intelligence how to use the important interaction between the possible inhibitor and the protein. So that's regarding our generative chemistry pipeline we have developed in cell medicine. So we got a very positive response, very positive feedback from various social media and the journals. Even the Wall Street Journal wrote about us. That here is Hong Kong-based company that is developing new algorithms to find new cure against COVID-19. So everyone is waiting for our new drugs, for our new effective medicine. But I would like to say that we will not get something final, something that we can use for humans. First of all, we need to find the compounds that could be effective and that can inhibit the main proteins. The next step is to find if these compounds inhibit the replication of the virus. So the whole virus, is it inhibited from the replication by this compound or not? The next step is to use some chimeric models of the mice that can just be infected by the coronavirus, and only after this long step process from just cells, mice, then monkeys, and only after that it will reach the clinical study and the humans. So the novel drug development is a very long and very cost effective process, but our main idea is to reduce the time from initial state of drug development. So as you could see, that we can develop new drugs just in few months instead of 2 or 3 years in the classical model. So let's wish us good luck. I will share with you our results when we published our results for drugs targeting, not the drugs, but the molecules targeting and inhibiting viral main proteins. So that's all for vaccine and drugs part of my session, of my speech. As it was presented before in the agenda, I would like to talk a little bit about the diagnostics of COVID-19. I'm not a clinicist, I am not a doctor, but I am a medicinal chemist, but I possibly can tell you something from my perspective of medicinal chemist about the diagnostics of COVID-19. So the main problem for the COVID-19 diagnostics is the test sensitivity and test specificity, and how to deal with the low sensitivity of the test. So the golden standard for COVID-19 diagnostic is a PCR analysis based on the genetic material of the virus. It's not very sensitive, it's very insensitive. It gets false negative results for about half of the cases, and but if it's very specific test, and if the if we got the positive results, it's always true positive results. But the problem is that we faced with a lot of false negative results. Another problem for a ELISA analysis based on the antibodies, it's both not very sensitive and not very specific test. But we need to deal with this. We need to use multiple kind of diagnostic tests. And another good idea is to use computed tomography of the chest, so we can diagnose bilateral coronavirus pneumonia in the late and middle stages using this technique. And the main idea is there is no ideal test. Every one test has maybe in some kinds very high specificity as PCR analysis, and in other cases you have middle level specificity and sensitivity, and the best approach is to combine all kinds of tests and methods to diagnose COVID-19. So to test, to increase the testing rate is the best idea to control and to prevent the disease. So that's, I think, all for our session. Thank you very much for joining us. If you have any questions, just put them into the chat. I will try to answer them about the COVID-19 vaccines, about especially for the drug development, because I am a medicinal chemist and we are mainly focused on developing new drugs. So, waiting for your questions. Please be free. So, regarding pneumococcal vaccine, that was asked in the chat. So, it was very controversial result from the northern Italy, because they used the pneumococcal vaccination just before the beginning of the pandemic situation in the northern Italy. You know, they struggles a lot with the pandemic. Very many deaths were in the northern Italy, in Milan, or in the Lombardia region. And in the autumn, they have a very big vaccination compound using this pneumococcal vaccine, and you know it was not very effective. But regarding other vaccines that I showed previously in the chart, they are studying pneumococcal vaccine, but it's not the mainstream for other. The mainstream, of course, are anti-tuberculosis vaccine and anti-poliovirus vaccine. Okay, so that's regarding pneumococcal vaccine, and for the severe infection of pneumonia, antibacterial drugs can be useful for treatment of super infection. So First of all, the viral pneumonia develops, and then some kind of bacteria can cause the superinfection, and that is why there is a broad usage of antibacterials, especially azithromycin and other um, very effective antibacterial agents. So it's not targeting the virus, it's not targeting the aggressive immune response, but the superinfection. And in the common um, methods of treatment of coronavirus, they of course use antibacterials to treat the superinfection. So, regarding the combination of azithromycin and HCQ, hydroxychloroquine, of course, it was known as a very popular from the beginning of a pandemic. It was developed of the French doctor from, I have forgotten, from Lyon University or something like this, or Marseilles University. But it's also very controversial because we do not know if the hydroxychloroquine or the azithromycin. So for the azithromycin, I think everything is clear. So it prevents developing the superinfection or the bacterial part of COVID-19. And for a HCQ, for the hydroxychloroquine and the chloroquine derivatives, it's still very controversial results. So you just can look at the clinical trials on the clinicaltrials.gov website to find out the the results of clinical studies, and you see, I do not want to commend these clinical trials because those are very controversial. I know there are very good positive results of usage of azithromycin, but I think everything is clear that just reducing the septal infection. So, any other questions? Please be free. Okay, so very interesting question about the smoking patients. So there is no good evidence that smoking patients are less susceptible to the coronavirus. Even there are some speculation that nicotine and SARS-CoV-2 use the same receptor. You know, the inhibiting of AC2 angiotensin converting enzyme receptor inhibiting cause hyperproduction of this receptor in the human cells. So if you inhibit something, the response of the organism will to increase the number of copies of the receptor. It's a known case for all kinds of inhibition. It's also it's also links to the problem of AC2 inhibitors that used to prevent hypertension. So some renin-angiotensin system inhibitors. You can see that the results for these inhibitors, example inhibitors of AC2 receptor, very controversial, and the vast majority of clinical doctors they reduce the usage of these inhibitors, and even the nicotine that could inhibit. It also can cause the hyperexpression of this receptor in the human cells. So um, this kind of receptor angiotensin that can be overexpressed using the nicotine, even it can inhibit. So I don't believe that the idea of nicotine inhibiting of human cells is a good idea. So I think there is no very good journal, for example, Lancet journal or the New England Journal of Medicine have published the results for smoking patients. They have no such evidence, and they have not published any article based on this idea. So they tried to use this captopril. So the all angiotensin converting enzyme inhibitors are ending with a pril suffix, and they tried to use this, but this treatment was not very effective. And nowadays, no one is used in the clinical studies. In the first stages in China, in February, in March, they tried to use these angiotensin converting enzyme inhibitors, but nowadays there is no attempt and new good results for this kind of treatment. So thank you very much for interesting and thoughtful questions. Akash, thank you. Thanks a lot. I think that could be very useful for other people looking and watching our presentation. So I think we have 5 more minutes for some of your questions. Oh, so thank you for plasma therapy. I think it's a very good idea using this plasma therapy, reconvalescent plasma therapy. I know there are very good results using this plasma therapy from the people who have suffered. But that the problem is to find a good concentration of antibodies, so in the plasma, and we need just very special techniques. So we do not have a good technique to find if there is enough antibody in the plasma from the post-COVID patients, but the results are very good. And it's some kind of a vaccine, because the vaccine could be a very similar, and it's very similar to the antibody therapy, because we can use just some some antibodies that can just kill just kill coronavirus particles. I think it's a very good idea, and I know that in the United States, it's the main therapy that saved thousands of people from dying. But the results are also not very clear, so we are waiting for a phase 3 results of using reconvalescent plasma. But I think it's a very good idea. It's an old known one, because from history of vaccination, there are multiple times when they used the blood cells and the blood plasma to prevent the disease. So I think that it's a very useful one, but unfortunately we cannot use it in some industrial way. So it's not very productive because we need first of all to find someone that have these antibodies in the enough quantity, and we can use it. We cannot use it just in a very productive way. But in the cases of pandemic, all kind of treatments need to be tested, and in this situation, especially in the big cities, if you have enough post-COVID patients, you can use it. But I think the best way is the vaccination, development of vaccines and drugs. So thanks a lot for joining us. Please be safe in this very hard situation. I will wish you and your relatives, parents, be safe, not to face the COVID-19. And good luck for everybody. Good studies. Thank you very much for joining us. Bye.